Hello and welcome dear students. Today we are going to discuss the key topic, that is cell fixation and staining. The objectives of our study lecture are to have general idea about cell fixation, to discuss about freeze drying and freeze substitution methods of preservation, to get an idea about embedding and microtomy techniques, to have a brief discussion on cell staining and types of stains, and to discuss the chemical basis of staining. Dear students, let us first of all discuss about fixation. Fixation brings about the death of cells in such a way that the structure of living cells is observed with a minimum of artifacts. The choice of fixative is dictated by the type of analysis desired. Fixation is a critical step by which the biological samples are preserved from decay, thereby preventing autolysis or putrefaction. The principal macromolecules inside a cell are proteins and nucleic acids. Fixation terminates any ongoing biochemical reaction and may also increase the mechanical strength or stability of the treated tissues. The broad objective of tissue fixation is to preserve cells and tissue components, and to do this in such a way as to allow the preparation of thin stained sections. In performing their protective role, fixatives denature proteins by coagulation, by forming additive compounds, or by combination of coagulation and additive processes. A compound that adds chemically to macromolecules stabilizes structure most effectively if it is able to combine with parts of two different macromolecules, an effect known as cross-linking. Now moving to the types of fixation processes. There are generally three types of fixation processes depending on the initial specimen. Heat fixation. After a smear has dried at room temperature, the slide is gripped by tongs or a clothespin and passed through the flame of a Bunsen burner several times to heat, kill and adhere the organism to the slide. Routinely used with bacteria and archaea, heat denatures the proteolytic enzyme and prevents autolysis. Heat fixation cannot be used in the capsule stain method, as heat fixation will shrink or destroy the capsule and cannot be seen in the stains. Immersion. The sample of tissue is immersed in a fixative solution of volume at a minimum of 20 times greater than the volume of the tissue to be fixed. The fixative must diffuse through the tissue to fix, so tissue size and density as well as the type of fixative must be considered. This is a common technique for cellular applications. Perfusion. Fixation via blood flow. The fixative is injected into the heart with the injection volume matching cardiac output. The fixative spreads through the entire body and the tissue doesn't die until it's fixed. In both immersion and perfusion fixation processes, chemical fixatives are used to preserve structures in a state both chemically and structurally as close to living tissue as possible. This requires a chemical fixative. Now moving to the various types of fixatives used in histology. Number 1. Cross-linking fixatives. Aldehydes. Cross-linking fixatives act by creating covalent chemical bonds between proteins in tissue. This anchors soluble proteins to the cytoskeleton and lends additional rigidity to the tissue. The most commonly used fixative in histology is formaldehyde. Formaldehyde fixes the tissue by cross-linking the proteins, primarily the residue of the basic amino acid lysine. Another popular aldehyde for fixation is glyceraldehyde. It operates in a similar way to formaldehyde by causing deformation of the alpha helix structures in proteins. Consequently, glyceraldehyde fixation on thicker tissue sample may be hampered, but this problem can be overcome by reducing the size of tissue sample. One of the advantages of glyceraldehyde fixation is that it may offer a more rigid or tightly linked fixed product. Its greater length and two aldehyde groups allow it to bridge and link more distinct pairs of protein molecule. Number 2. Precipitating fixatives. Precipitating or denaturing fixatives act by reducing the solubility of a protein molecule and often by disrupting the hydrophobic interactions that give many proteins their tertiary structure. The precipitation and aggregation of proteins is a very different process from the cross-linking that occurs with the aldehyde fixative. Number 3. Oxidizing agents. The oxidizing fixatives can react with various side chains of proteins and other biomolecules, allowing formation of crosslinks that stabilize tissue structure. However, they cause extensive denaturation despite preserving fine cell structure and are used mainly as secondary fixatives. This group includes osmium tetroxide, potassium dichromate, chromic acid, and potassium permanganate. It has a binding effect on protein similar to that of formalin. Following fixation with potassium dichromate, tissue must be well washed in running water before dehydration. It is used for fixation of fatty tissues and nerves. Number 4. Mercurials. Mercurials are mercury-containing fixatives which increase staining, brightness and give excellent nuclear details. It includes B5 and Zenker's fixative. Despite being fast, they penetrate poorly and cause tissue shrinkage. Number 5. Picrates. They penetrate tissue well to react with histones and basic proteins to form crystalline picrates with amino acids and precipitate all proteins. Dear students, now moving on to the another objective, that is freeze drying and freeze substitution. Freeze drying method. This method consists of rapid freezing of tissues followed by dehydration in a vacuum at a low temperature. The initial freezing is generally accompanied by plunging small pieces of tissue in a bath cooled with liquid nitrogen to a temperature of -160 to -190 degrees Celsius. Fixation in liquid helium at 0 Kelvin has also been used. The tissues are dried in a vacuum at -30 to -40 degrees Celsius. Under these conditions, the ice in the tissues is changed directly to a gas and dehydration is achieved. Advantages of freeze drying method. The tissue does not shrink, fixation is homogeneous throughout, soluble substances are not extracted, the chemical composition is maintained and the structure in general is maintained with very little modifications, and fixation process is very fast. Now, freeze substitution method. Freeze substitution was described by Simpson in 1941 as an excellent method of preparation of tissues for study in microscope. It is based on rapid freezing of tissues followed by solution substitution of ice at temperature well below 0 degrees Celsius. The frozen tissue is placed in substituting fluid at -70 degrees Celsius for about a week to dissolve the ice slowly without distorting the tissue structures. Excess solution is washed out and the specimen is embedded, sectioned and stained conventionally. For best morphological and histochemical preservation, substituting fluids in general contain both chemical fixing agents and solvents for ice. For example, 1% solution of osmium tetroxide in acetone, mercuric chloride in ethanol, and picric acid in ethanol. Specimen size should be up to 3 mm in diameter. In the freeze substitution method, the tissue is kept frozen at a low temperature -20 to -60 degrees Celsius in a reagent that dissolves the ice crystals, ethanol, methanol or acetone. Rapid freezing is generally used for preservation and storage of cultured cells for later recovery and plating, also used for preservation and later recovery of viable sperm for artificial insemination as well as fertilized eggs. Dear students, moving on to the next part, that is embedding. Embedding is a process in which tissues or the specimens are enclosed in a mass of embedding medium using a mold. Since the tissue blocks are very thin, they need a supporting medium in which the tissue blocks are embedded. This supporting medium is called embedding medium. Various embedding substances are paraffin wax, celloidine, synthetic resins, gelatin, etc. The choice of embedding medium depends upon the type of microscope, type of microtome and the type of tissue. Paraffin wax with high melting point is used for embedding. The molten wax is filtered inside the oven through a filter paper into another container. Carbo wax is a water-soluble wax, therefore the tissues are directly transferred to water-soluble wax after fixation and washing. Epoxy resin is used in higher resolution work and to see greater details and is used for electron microscopy. Agar embedding is mainly used in double embedding. Celloidine embedding, celloidine in purified form of nitrocellulose and is used for cutting hard tissues. Dear students, now we will discuss microtomy. After successful embedding, sectioning of specimens is done by microtomes. Microtome, micro means small and temnin means cut, is a tool used to cut extremely thin slice of material known as sections. Earlier microtomes enabled free-hand sectioning of fresh or fixed material using a sharp razor. Modern microtomes are precision instruments designed to cut uniformly thin sections of a variety of materials for detailed microscopic examination. Central to the production of good sections is the microtome knife. A sharp knife edge free from imperfections is essential for the production of good sections. Various types of microtomes include hand microtome, slash microtome, cryostat, saw microtome, rotary microtome, and ultra microtome. In slash microtome, sample is placed into a fixed holder which then moves backwards and forwards across a knife. Rotary microtome is a common microtome design, operates with a staged rotary action such that action cutting is the part of the rotary motion. Cryomicrotome is used for cutting frozen samples. Tissues should be conveniently sectioned before they are observed under the microscope. For this purpose, freezing microtomes cooled with liquid carbon dioxide are frequently used. Instruments consisting of a microtome enclosed in a chamber at low temperature, the so-called cryostat, can make sections of fixed or fresh tissue for cytochemical purposes. Dear students, moving on to the next part, that is staining, types and chemical bases. Cell staining is a technique used for the main purpose of increasing contrast through changing the color of some of the parts of the structure being observed, thus allowing for a clear view. There are a variety of microscopic stains that can be used in microscopy. Staining of sections for the light microscope is necessary because most parts of the tissue is colorless and the various elements cannot be distinguished. Chemistry behind all dyes is acid-base reactions. Molecules such as nucleic acids have an affinity for basic dyes, and basic regions of the cell or some proteins have an affinity for acidic dyes. Vital staining refers to the uptake of dyes by cells. Number 1. Types of staining. There are two types of staining techniques: in vivo staining and in vitro staining. In vivo staining refers to the staining of a biological matter while it is still alive. In vitro staining refers to the staining technique where the biological matter is non-living. Most cytological stains are solutions of organic aromatic dyes. Two common types of dyes have been recognized since the times of the pioneer work of Ehrlich, which includes acidic dyes and basic dyes. Number 2. Various types of stains used in histological examination includes hematoxylin and eosin staining. These are two stains used in the examination of thin slices of biological tissue. Contrast is created by the stains where hematoxylin turns the nuclei blue while eosin turns the cytoplasm as well as other parts pink or red. The hematoxylin is a basic dye that stains acidic components of cells a blue color. Hematoxylin stains the nuclei of cells and the RER of the cytoplasm. Eosin is an acidic dye that stains the basic components of the cell a reddish pink color. This characteristic is known as acidophilia. Most of the cytoplasm of cells is stained by eosin. Bone matrix is also stained by eosin. Number 2. Periodic acid shift staining. Periodic acid shift staining is widely used staining technique that stains the neutral sugars of glucosamine glycans a pink color. Common components stained positively with PAS includes mucus, glycogen, mucoprotein, glycoproteins, the basal lamina, organ capsules and blood vessels. Number 3 is osmium tetroxide. Osmium is used to stain lipids a dark black color. It is very useful for demonstrating the myelin of myelinated nerves or lipid droplets in the liver or steroid secreting cells. Number 4. Giemsa stain. There are a variety of Romanowski type stains with mixture of methylene blue, azure and eosin compounds. Among these are the Giemsa stains and the Wright stain or Wright's Giemsa stain. The latter is utilized to stain peripheral blood smears. Number 5 is gram staining. This is one of the most common staining technique. It is largely used to differentiate bacteria species as either gram-positive or gram-negative. This is achieved through the chemical properties of bacterial cell walls, where different colors are displayed after staining. This technique is based on the fact that the gram-positive cell wall has a strong attraction for crystal violet following the addition of iodine as compared to the cell wall of gram-negative. Iodine is the mordant and forms a complex with crystal violet, which is easily washed off from the gram-negative cell wall using ethyl alcohol. Now moving on to the next point, that is chemical basis of staining. In a basic dye, the chromophoric group which imparts the color is basic cationic. For example, methylene blue is a chlorohydrate of tetramethylthioneine. in which the basic part carries the blue color. Eosine is generally used as potassium eosinate, in which the base is colorless. Sometimes the two components of the salt are chromophoric, for example, eosinate of methylene blue. The most frequently used chromophore for acidic dyes contain nitro and quinoid groups. Basic chromophores contain azo and indamine groups. For example, picric acid has 3 nitro groups and 1 hydroxyl group, also called oxochrome, by which the dye combines with the tissue. The properties that enable protein, certain polysaccharides and nucleic acids to ionize either as base or acid should be noted. Acid ionization may be produced by carboxyl, hydroxyl, sulfuric or phosphoric groups. Basic ionization results from amino and other basic groups in the protein. At pH values above the isoelectric point, acid groups become ionized. Below the isoelectric point, basic groups dissociate. Because of this property, at a pH above the isoelectric point, proteins will react with basic dyes, methylene blue, crystal violet or basic fuchsin, and below it with acidic dyes, orangey, eosine or aniline blue. The net charge of nucleic acids is determined primarily by the dissociation of the phosphoric acid groups, and the isoelectric point is very low, that is pH 2 or less. For this reason, staining with basic dyes at low pH is selective for nucleic acids. Dear students, now let us have a brief idea about metachromasia, a change in original dye color. Basic dyes belonging to aniline group differentiate particular tissue components by staining them a different color to that of original dye by phenomena known as metachromasia. It occurs due to polymerization. Metachromasia takes place when certain negatively charged groups on the tissue react with cationic dye. On polymerization, the original color of the dye changes to another color. For example, mast cells stain pink with toluidine blue. Thionin and toluidine blue dyes are commonly used for quick staining of frozen sections using their metachromatic property to stain nucleus and cytoplasm differently. Metachromasia is enhanced when intermolecular distances are reduced. Metachromasia has interesting histochemical and physicochemical implications. The reaction is strong in cells that contain sulfate groups, for example, the cells of connective tissue. Factors which enhance metachromasia are: number 1, increasing the concentration of dye, number 2, decreasing the temperature, number 3, pH, and number 4, water. In tissues where there is high concentration of anions, the cationic dye molecule may be held in such close proximity to one another that van der Waals forces can exert their influence and cause dye to polymerize. Consequently, the color changes from blue to red. With this, we come to the end of today's program. Hope you have enjoyed it well. Thank you for watching.